Next question comes from Soul Flayer. This is question number 2 from Soul Flayer. This is it's one of my favorite names. Could you dive a little deeper into yeast repitching and any links to so-called house strains and the impact, if any, of repitching the same yeast over and over and over again? I did a little research on this one. I listened to somebody recently who was saying, oh, they had repitched their yeast, like they've been brewing for 15 years and they're still on the same the original pitch, right? And people talk about house flavor, different things like that. Let's dig into this a little bit. This idea of using a yeast strain over and over and over, and just allowing it to kind of because it will develop to your processes, because really what you're doing is you are self-selecting that strain for your exact process, for your ingredients. So there will be changes over time. So let's dig into this. Respiratory deficient mutants, also referred to as RD mutants, are defective yeast cells. All yeast used in brewing have the ability to ferment or respire specific carbon sources. Alcohol is only produced when sugars are fermented. However, respiratory ability too appears to be important for yeast's ability to produce acceptable beers. RD yeast mutants are also called petite mutants, or French for small, because of their small colony size when they are propagated on an agar medium. Medium. These mutants show sublethal damage to their mitochondrial DNA. So there's damage to that mitochondrial DNA, but not to the point to where it will kill. They occur more frequently than nuclear mutants because of their proximity to the highly oxidative electron chain. I don't know what that means. Importantly, they lose their ability to respire and use carbon sources such as ethanol or glycerol, but they can still ferment, albeit slowly. Now let's start getting into the money here. There are 2 forms of RD mutants, each differing in the nature of their DNA damage. Rho mutants contain incomplete mitochondrial DNA, whereas Rho mutants, I don't know, do not contain mitochondrial DNA at all. Only Rho mutants are observed in brewing, and they occur spontaneously in concentrations of up to 4% depending on yeast strain. Increased mutation rates can develop if brewer's yeast is stressed or not handled properly during harvest and storage. Mutations can also accumulate during cereal pitching. A culture can contain high levels of RD mutants of perhaps 10%, and that can affect fermentation performance as well as produce noticeable off flavors, particularly the clove-like 4-vinyl guaiacol. I didn't never pronounce that one right. RD mutants are easily detected in brewery lab tests, and high concentrations will spur a brewery to reculture the yeast strain to arrest the development of further yeast health issues. So, big picture, we can form these petite mutants as we're going generation after generation with our yeast. The thing that are going to keep your yeast happy, and this is going to be different probably for every yeast strain, exactly how they prefer to be handled. As far as storage time, the amount of time they can handle being down in storage, not actively fermenting work, making sure that you're keeping up on appropriate levels of nutrition, making sure that you are being very, very clean on the lab side, on the harvest side, on the repitch side. You need to be really smart with this stuff. I think you're playing games that maybe aren't worth it if you just keep trying to serial repitch something and you don't have the necessary SOPs or cleaning capacities and or lab capacities in order to carry that out. Because as you can see, these petite mutants can continue to grow, and it only needs to make up about 10% of that pitch in order for you to have some delayed fermentations, this, that, and the other thing. Okay. So that's kind of my pitch on that, man. I think it's a great question. This is another one that makes me do a little digging on, and I appreciate it. That was fun to check out.